Okay, so yesterday we started considering some of the some of the biochemistry of muscles, and today we will consider further all the biochemical details that we are able to compress into one lecture. Uh, so first of all, I want to consider a little bit about muscle energetics. So where do we get energy for the muscles to work, and how do we get that energy? Of course, we will not be able to consider the full pathways from whatever we have eaten up to the endpoint, that is the ATP. I hope you remember that we require ATP for the contraction. So we have considered yesterday the actin myosin interaction, so they need ATP. And there is another very crucial point. We need ATP for calcium pumping. So if we move calcium around the cell, especially between the cytosol and the reticulum, we will need all that pumps to move calcium ions, and of course they require ATP, because it does not it does not just move on its own. You need ATP to move that ions inside of the sarcoplasmic reticulum. Okay, but at least certain points of the muscle energetics we will be able to consider, and let's do it. First, a small notion regarding the efficiency of the muscles. It turns out that the percentage of the input energy to the muscle in form of chemical energy and nutrients can be converted into work even under the best conditions, with the efficiency that is less than 25%. So we are pretty ineffective in this sort of when we think about our muscles from this kind of standpoint. Of course, we have certain machinery that is much more effective than we are if we consider some conversion of energy. The most effective conversion of energy that into mechanical work that we know is probably the conversion of the electrical energy. So electric motors can be 99% effective. If we consider some engines that are based on transferring heat, so some steam engine or some different heat engines, they rarely go beyond 40%, and we are something around 25%. So that's basically our positioning in the world if we compare ourselves with our creations, our machinery. The reason for this low efficiency is that something like 1/2 of the energy in the foodstuffs is lost during the formation of ATP, and even then only 40 to 45% of energy in ATP itself can be later converted into work. The effectiveness depends on the contraction velocity. If we are using some very slow contraction, the thing is that chemical reactions, they do not stop. Even if we do not do any efficient work, like I'm just holding my hand and you know that I'm not changing anything, it just hold it still, I'm still using up some energy, because chemical reactions are going and going, and I'm losing that energy in form of heat. So it is dissipated into the environment in form of heat, and that's why it turns out that it leads to a very low efficiency. The best efficiency we get during some sort of moderate velocities, so that's maximal efficiency that can be up to 25%. And if we make rapid contractions, then a lot of energy will be used to overcome certain viscous friction within the muscle itself. So we are viscous, so when we are moving and when we are moving fast, we need to spend some energy to overcome that viscous friction. Here are some sources of the energy for muscle contraction. Well, as I told you, we need it for, first of all, that cross-bridge cycling, so that's the actin-myosin interaction we were considering yesterday. We need it for calcium reuptake in the sarcoplasmic reticulum. That requires a lot of energy. And of course, we need ATP for sodium-potassium pumping. Well, humans' organism uses some incredible, I guess, amount of energy for all that pumps to work. So we spend a lot of energy from the foodstuff we eat just to move the ions around. So that really matters. So what kind of sources of energy do we have? So the first thing, the first and the foremost thing that is used, that's of course ATP. So all the reactions we have considered, they use ATP. If we are trying to use something different, it should be first converted into the ATP. So that's pretty simple, pretty reasonable. Of course, in the cell, there is some small storage of the ATP, so something around 4 micromoles. That's good enough for some contraction, for 1 or 2 seconds contractions. So if you are doing something pretty quickly, so that lasts for 1 or for 2 seconds, ATP in the cell would be pretty enough to perform that action. Of course, we are not moving just for this brief period of time. We need to perform some more prolonged actions, and we will need some other sources that will help us to replenish the ATP that we have used up. And these are 3 systems, and some of them we will consider in the next slides in a little bit more details. So what are these systems? First of all, the phosphocreatine system. The phosphocreatine stores high energy phosphate bonds and transfers phosphate to ADP to regenerate ATP instantly. The amount of the phosphocreatine that we have usually will be enough to supply like 5 to 8 seconds of maximal effort. So we can use up our creatine that we have to generate ATP and to use then this ATP, and that will be enough for 5 to 8 seconds of do something. That may sound pretty small amount of time, but this immediate energy, these 5 to 8 seconds, that's pretty enough to run a sprint. So I hope you remember the world record for, I don't know, for 100 meters. That's the numbers will be somewhere around 8 seconds. So you can make a 100-meter sprint basing on the energy stored in the phosphocreatine system. So despite it looks really small, that may be enough to do something. Maybe if you're running away from some predator, that may be pretty handy, let's say. So that's why we may need this sort of system. The second one I will spend a little bit more time on is the glycolytic system. So you have probably heard the term glycogen, and this glycogen can be stored. It is stored in muscles, and it is stored in the liver. The storage in the liver, I think we will consider in the next lecture with more details. That's some sort of pretty interesting source of energy, like some battery for the whole organism. But except for that storage in the liver, we have a pretty handy storage in our muscles. So the breakdown of that glycogen that works without oxygen, so that's under the anaerobic conditions. That's pretty handy. It gives you 2.5 faster ATP conversion than oxidative metabolism. That's pretty good, and that may support your activity up to 1 minute. So if you are doing something under the so-called anaerobic conditions, And I remind you that anaerobic condition, it doesn't mean that you don't breathe. Uh, you breathe, but there are certain kinds of activities that you are doing something so vigorously that the blood has no time to supply enough oxygen to your muscle. So they are starved of oxygen, so they are depleted of oxygen just because blood cannot supply it in such amounts that are needed to perform all that actions and to get all the needed energy. So please don't mix these things up. So anaerobic conditions means that you are just doing something very vigorously, and blood cannot supply enough oxygen. It doesn't mean that you are holding your breath or that you are doing some activities underwater. So no, it's not. And of course, the third pathway, that's oxidative metabolism. We have already considered that. That's whatever has to do with the Krebs cycle, with the electron transport chain, mitochondria, and so on. So we had the whole lecture on that, and I hope that you remember all the things. They are pretty similar in all different tissues. So whatever you know is applicable to the to muscles as well. Good. Here's once again all these three systems that show you where do we get energy. So despite the ATP that we already have in our cells, but there's only a small amount of that, you can gain some more ATP from phosphocreatine, glycogen, or with oxidative phosphorylation. We considered how the glucose is transformed into energy when we get through the glycolysis and then Krebs cycle. But I guess in the next lectures we will consider a little bit more details about fatty acids entering the Krebs cycle and amino acids entering the Krebs cycle. So we will consider that, I guess. Here is a small table just for reference. So how much time do you have basing on each of these systems? So the phosphocreatine system, the glycogen lactic system, and so on. And here are different forms of activities. Once again, you can take a look after the lecture just for a reference. During which activity which of these systems are used. And of course, as the rule of thumb, you are first using your ATP, then phosphocreatine, creatine, then glycogen, and then you switch to the aerobic system. And here you see that the shorter the duration of the activity, like 100-meter dash, then you are switching to different systems. So for for aerobic system you have something like marathon run or jogging, while for 100-meter dash that's enough just the phosphocreatine system. And here is one more nice picture that shows all these systems in one slide. What else? This picture I hope you have already seen. These are Cori cycle and Cahill cycle. So one of them, here you see glucose and pyruvate. We have already considered that when we were talking about the glycolysis. So you remember when we have an anaerobic pathway of processing glucose, we end up with the phosphoenol pyruvate, and then we need to do something with the pyruvate and somehow regenerate when we need some NAD plus or NADH, whatever. So we have considered that part. So we need some regeneration in the NAD plus and the NADH system to return to the balance, and we need to perform certain reactions on this pyruvate. Depending on the organism, these reactions may be a little bit different. So we in our organisms, we process pyruvate to the lactate, then the lactate can be exported to the blood. So from the muscle it can be exported to the to blood, then from blood it is imported into the liver. In the liver it can be reconverted to the pyruvate, and then by gluconeogenesis it can be converted back to the glucose and returned to the bloodstream, and then it can be reuptaken by different organs, including the muscles. So we have considered this part, and the other cycle is processing to the alanine and then converting the alanine to the pyruvate and then back to glucose, and so on. So these are just two different cycles. So glucose-alanine cycle is important in condition of starvation. Thus, net transfer of amino acid nitrogen from muscle to liver and corresponding transfer of glucose from liver to muscle is affected. And alanine cycle, as you see here, it's intimately related to the Corys cycle. Here is example how the creatine phosphokinase works. So here we have creatine. So of course I should show you these sort of formulae. That's biochemistry. So here we have ATP, and here we have phosphocreatine and ADP. So depending on the condition, we can shift this equilibrium to the left or to the right. So if we have conditions where we need to perform some work and we need energy, we are shifting things to the left. So we use up phosphocreatine to get ATP. So we are transforming ADP into the ATP. If we are under conditions that we are relaxed and we have some time to replenish our storage of the phosphocreatine and maybe some other chemicals, we are shifting everything to the right. So we are synthesizing ATP, let's say, by the glycolysis pathway and Krebs cycle and electron transport chain. So we use that, we synthesize ATP, and we can use that ATP to transform creatine into the phosphocreatine. Here you see this sort of scheme, how the creatine phosphokinase works. So it binds to the ATP and creatine independently on the on how do they bind, who the first and who the second one, independently on the order. And here you see that they can leave this enzyme independent on the of the order as well. So we have seen these sort of pictures, and I hope you remember there are different enzymes, and some enzymes, they respect certain order. They want certain chemicals to be bound in some predefined order, while the other enzymes, and this one, they don't care. So who binds, that's good. Here's pretty interesting image for you. This is once again the scheme where the phosphocreatine and ADP during activity is transformed into the creatine and ATP, and during recovery transformed back. And interestingly, we can see that almost in the real time. So if we do a nuclear magnetic resonance spectrum of 31 phosphorus, we can see, so I hope you remember our lecture, I guess either number 2 or 3, something like that, where we were considering all that nuclear magnetic resonance things. And we have seen that there is some sort of chemical shift depending on the environment of the phosphate that we have. And here is the plot. That's basically that resonance plots, but they are stacked one after another with these coordinate being time. So this is time, and here we have that sort of NMR plot. And if we consider these sort of NMRs, many different NMRs, and we know that there was a period of rest and period of exercise. So this is the period of exercise. So these plots, they show the period of exercise. And you might see in let's say in real time, how the phosphocreatine, the amount of the phosphocreatine and the inorganic phosphate, they change in the cell. So here you see phosphocreatine, there is certain amount of it. Here you see ATP. Here the tricky question for you. Why 1 peak for phosphocreatine but 3 peaks for ATP? Pretty simple question, just to check you're not sleeping. Three mole of ATP is produced from one. I'm not sure what you mean. What does the nuclear magnetic resonance 3 phosphates? Yes, ATP has 3 phosphates. There are 3 phosphorus atoms and they are in different chemical environment. That's crucial as well, because if we have I don't know 100 atoms but they all are in the same chemical environments, we will not see them separately on these plots. You can remember that we have 10 to the 23 molecules in there, so there is there are many molecules, but we don't see 10 to the 23 peaks. Yeah, so we see only these 3. So yes, ATP had 3 phosphates in different chemical environment. They are seen here at 3 peaks, while phosphocreatine, if you look at this plot, you clearly see 1 phosphorus, and that's all. So it has only 1 phosphorus, so it can't give you more than 1 peak, so it gives you 1 peak. Here we have a peak for the inorganic phosphate. That's the phosphate group that was removed from the from the ATP during some processes. It contains only 1 phosphorus, so 1 peak, and ATP has 3 peaks because 3 phosphorus in 3 phosphorus atoms in different chemical environments. So that's good. And if you remember, this sort of NMR can tell you something regarding the number of that atoms, some relative numbers of that atoms. And here you see that the amount of ATP is more or less held to some up to some level. So while the phosphocreatine goes down during the activity, so we are using that phosphocreatine to perform some mechanical work, then we have some recovery and phosphocreatine gets back up. And here we see the inorganic phosphate that was removed during operation. It goes up when the phosphocreatine goes down, and it goes down when phosphocreatine goes up. Well, that's pretty reasonable because we are using that to phosphorylate back the phosphocreatine. So yes, so as you see, we can use NMR even in such cases, and we can gain some insights of what is happening with the chemical composition of some living, I don't know, some preparate, or maybe that was some living muscle. Good. Just few more images for you regarding this stuff. Here is the effect of diet on the rate of muscle glycogen replenishment. So when we are getting to the glycogen, I guess yeah, I guess we should get now to considering the next level, the glycogen. So suppose we have used our phosphocreatine, and now it's time to switch to the glycogen and get energy from there. And here you see how that glycogen is replenished based on the diet. And here you see the high carbohydrate diet that, of course, should be good for replenishing the glycogen, because we use glucose to build the glycogen. So you may guess from the names that they are somehow connected, and I will show you in the next slides how exactly they are connected, what are the chemical formulae, and what's they're so interesting. Here are some more interesting pictures. These are world track records that depend that basically gives you some idea regarding dependence of the running time and the velocity, the mean velocity that people may handle. And here you see that these plots, they show you that the longer the distance, the smaller is the mean velocity that we can handle during that distance. So that's once again that that hints you that we have some different mechanisms, and some mechanisms are turned on when we are doing some mechanical work pretty vigorously, but we can do that for a prolonged period of time, while the other mechanisms are able to sustain some activity during pretty long time intervals, but we can not do that activity so vigorously as we do that on the small time scales. Good. Regarding the glycogen, so here you see this sort of funny picture. That's it. The core is this sort of yellow thing. That's the core. It's some sort of protein called glycogenin. Each line represents glucose molecules joined by the alpha 1,4 glycosidic linkage, and the non-reducing ends of the glycogen molecule form the surface of the glycogen granule, and degradation it takes place at this surface. Here you see the formulae, so you see a lot of glucose units that are connected with these alpha 1,4 linkages. So if you see the numbering of the carbon atoms in the in the glucose, you might guess where do these numbers come from. So this is carbon 1, here is the carbon 4. You remember that glucose has 6 carbon atoms, so the next one will be the 5th carbon atom, and this one in CH2OH, that's the 6th one, so the last one, carbon atom. So these are the linkages. There are, of course, some linkages alpha 1,6. They are created with the separate ferment and separate enzyme, and I guess I will show you these all these enzymes, how they work, just in a minute. What's interesting, first, you see these sort of ends. These are called the non-reducing ends. What so interesting do you see here? Why do you think they want to be oriented towards the surface of the glycogen? No? What's water? Water is a polar solvent, yeah, so HOH, so that's a polar solvent. So it really likes some polar molecules and polar groups. So these sort of OHs, so here you see a lot of OH groups, they will be pretty welcome in the water or water-like environment. So if you have some polar solvent, it would like to see these sorts of groups. So if you will have some large molecule, and that molecule will have different on its different ends, different groups, So the polar groups like OH groups, they will tend to be oriented towards the solvent, while the other things will try to hide somewhere. That's a pretty similar thing that you see with phospholipids if you consider a membrane or something like that. So you remember that lipid bilayer. So in a lipid bilayer that is constructed from the phospholipids, once again we have some polar groups. These polar groups are oriented towards towards water, and they are trying to hide all that hydrophobic thing inside. So here is a pretty similar situation in a sense that here you have many polar groups, and of course they want to be oriented towards the surface. What else is interesting? Um, if what if we try to store our glucose in form of free glucose? So why do we need all that glycogen thing? So why we are lumping all these glucose together instead of storing that in a form of the glucose? So here I want to make you a small hint. Do you remember what is osmosis? Osmosis. Osmotic pressure, maybe. Um, and can you tell me why do we may want to collect all that glucose into something unitary? You remember the experiment? I hope you have seen that maybe anywhere. So here I have a tube, and here is let's say some membrane, and outside the tube I have, let's say, some water. So I have water here. So that's water. And here inside the tube, water can go inside, can go outside, but inside the tube, except for the water, I will place I don't know, some ions of something. For example, so just, I don't know, some salt. So here they are. What will happen to the level of the water inside the tube? Who remembers from, I don't know, any physics course you may have taken? Yes, it will increase. So the water level will increase inside the tube. So we have some additional so-called osmotic pressure. So molecules of water are getting through the membrane. So this level will increase because molecules of water will come through the membrane and try to dilute whatever we have inside of that tube. And the situation is pretty similar for the cells. If you have something inside the cell and the concentration of that is pretty different from what is outside the cell, the water will try to come in the cell and dilute whatever there is. And I'm pretty sure if you ever had some labs, I don't know, maybe in botany or something like that, you should have seen these sort of effects on the cells, on the plant cells. So when you take some plant cells, you put them under the microscope and you are dripping some water, and that water may either contain a lot of salt or contain no salt or something like that. And you may see like that cells, they are shrinking or they are increasing depending on the concentration of these salts around the cells. So you may even see how they come off the cell walls when they are shrinking or when they are when they are bulging, and you see that pressure inside is too large, and of course that may even come to the point when the cell will be destroyed. So that's pretty possible. So you don't want this sort of situation. And the thing is that if you calculate all that stuff, you will see that these sort of effects, they depend on the number of molecules that you have in here. So not on the size of that molecules, but on their number. So that comes from that entropic part and so on. And the idea is that if you don't want that sort of osmotic pressure, you think that it's not good for the cell, it may destroy the cell, it may be a good idea to collect many molecules into one molecule, and that will lower the osmotic pressure, and that will be good for your cell. At least it will not explode. So good. But of course, that brings us a lot of trouble regarding processing all that glycogen, and we will need a number of different enzymes to create glycogen or to destroy it. So depending on whether we need energy or we are trying to store it. But anyway, maybe that was the only way to to work around the osmotic problem. So maybe it's has evolved this way because of the osmosis. Good. So regarding the structure, so that's large branch polymer, like 55,000 glucose residues, can be up to 40 nanometer. Pretty close to starch in sense of chemistry and so on. Contains mostly alpha-1,4 glycosidic bonds with alpha-1,6 branches every 12 residues. Branching, of course, helps us with rapid mobilization and solubility, because when you have these branches, first of all, you have more ends to process if you need to get your glucose. Otherwise, you will need to chop off just one molecule of glucose each time, and here you have many ends, so you can process them simultaneously. And of course, you have if you have many ends, you have many these sort of OH groups, and that's much better for the solubility, as I have explained. Present in bacteria, archaea, and eukaryotes. Plants store glucose as starch, but it's pretty similar chemically. Maybe at some point we'll get to that, maybe not, I'm not really sure. Reactions. If we have glycogen stored, of course we need to somehow break it down to get our energy out of it. So it's called the phosphorylases. Maybe a strange name for you, but you have already heard from me the word hydrolysis. So hydrolysis meant that we were using water to break something down. So we were expecting some new HOH groups to appear. Here we use this sort of phosphate. Here you see HPO4 2 minus. And we get these sort of two molecules. So this was the glycogen N residues. So this is one of the ends of the glycogen. So these R means that we have some large molecule out there. So we are working just with one end here. And here we have separated one part of it and transformed into glucose 1-phosphate, and the rest of the glycogen N-1 residues are left. So the key enzyme is glycogen phosphatase. It cleaves glycogen by adding orthophosphate instead of water and produces glucose 1-phosphate. The reaction favors breakdown in vivo, since the proportion of concentrations in organic phosphate and glucose 1-phosphate is much greater than 1. I hope you remember that we can shift equilibrium according to the Le Chatelier principle, basing on the concentrations. And we already have seen even some reactions that have pretty high energy demands, let's say so, to overcome the transition state, but they are still driving forward, basing on these sort of concentrations. If you don't remember, you can check our lecture on Krebs cycle. We had few of them there, definitely. Oops. No ATP is needed. We get our sugar already phosphorylated. That's pretty good, because if you remember, why do we need glucose? We can use it in the glycolysis, and then whatever in the glycolysis we will get two ATP molecules as the payoff. But you remember that in glycolysis we started with glucose, we phosphorylated it, so we already we lose one ATP molecule for that. Then on the later stages we lose one more, and then we have four molecules as payoff. But here we have our glucose already phosphorylated, so that's pretty good. The second thing, phosphorylated glucose cannot leave the cell, so we remember that the first stage of glycolysis was to catch that glucose inside the cell, so this phosphorylation was needed. So that's pretty good. And what else? The mechanism, it works on non-reducing ends, so free OHs of the 4th carbon atom. The inorganic phosphate attacks glycosidic bonds and cleaves the bond between C1 and oxygen. The alpha configuration at C1 is retained, and then phosphoglucomutase converts glucose 1-phosphate into glucose 6-phosphate. Because we need glucose 6-phosphate to perform our reactions, but here we have glucose 1-phosphate. But that's pretty solvable. Here you see a reaction where we have our glucose, and here we have inorganic phosphate, and here we are trying to cleave these bonds. So we are trying to remove one glucose unit from the from the glycogen, and here we have the stages through which reaction goes. So it goes through the stage with the carbonium ion. So here we have carbonium ion. It uses pyridoxal phosphate covalently bound to the enzyme, and this pyridoxal phosphate, that's a derivative of the B6 vitamin. So that's one of the reasons why do we need vitamins. So one of them is pyridoxal phosphate that is involved in the glycogen degradation and giving us some energy. And here you see the result of this reaction. So phosphate group, here you see PLP, phosphate group, another phosphate group, and here how that goes. Good. What else? Glycogen phosphorylase is regulated. So of course the glycogen degradation is tightly controlled. Since the key enzyme is glycogen phosphorylase, it is highly regulated. So it may be regulated by allosteric effectors like energy state, or through reversible phosphorylation that is controlled by hormones. There are two different isozymes. One is more living in the liver and helps to maintain the blood glucose. I guess in the next lecture we will consider this part. And the other one is the muscle isoform. That's of course for our today's lecture to produce the contraction. Phosphorylase forms, there are two different interconvertible forms, phosphorylase A and B, and they may exist in two stages like R relaxed and T10 state. So here are different forms of the phosphorylase. Here are once again two different isozymes, so muscle and liver isoforms of the phosphorylase. I'm not sure if we need to get into these sort of details. let me check what I want to show you. Maybe maybe let's focus on this slide, and I will return as needed. So here is a signaling cascade of what is happening to start all that sort of reactions. So we have already considered some of the phosphorylating cascades and signaling pathways. So here you see we have a receptor depending on whether we are in the liver or in the muscle. So in the muscle, that's mostly controlled by the epinephrine. So here you see the formula. In the liver, that will be controlled by glucagon. Here it is in the top right corner. So that's a small peptide. So anyway, receptor is activated. Here you see this sort of thing, and you might guess what type of the receptor is this. So here's a tricky question for you. So what is this type of the receptor? Basing on the image that you see. Let's remind ourselves some types of the receptors that we know. So we know ion channels, so they can react to something and open or close. We know G protein coupled receptors, so receptors coupled to G protein. We know receptor tyrosine kinases, so receptors with some sort of kinase activity. And suppose for now these are the main classes, and this one falls into one of these classes. How do you think which one is that? G protein. Yes, absolutely correct. That's a G protein coupled receptor. So here you see the G protein. I hope you remember it has 3 subunits: alpha, beta, and gamma. Upon activation, the exchange of GDP for GTP is performed, and then this sort of alpha subunit may go away and activate something. Something may be adenylate cyclase that creates a very, I would say, famous secondary messenger. That's the cyclic AMP. So it takes ATP and creates cyclic AMP, and this cyclic AMP may activate something else. And in exactly this cascade, it activates the protein kinase A that activates the phosphorylase kinase that transforms phosphorylase B into the form of phosphorylase A. So if we need some energy, that's basically the pathway that is performed. And here is a short reminder for you, why do we exchange here GDP for GTP if we may have, I don't know, already contain some GTP inside of this G protein? What is the logic of that? What happens to that GTP later? Yes, absolutely correct. That's a way to turn off So we don't want our cascade to work indefinitely, so we need something to shut down that sort of cascade. So of course there will be some additional things that turn off all that signaling, but one of such switches, that's basically that GTP. So after some time passes, the GTP will be hydrolyzed to the GDP, and that alpha subunit will just switch off and will stop activating whatever it was activating. In this case, that was for production of the cyclic AMP with the adenylate cyclase. So here you see how phosphorylase B is transformed into phosphorylase A. A small reminder that in muscle, so we see there's some difference between liver and muscle. So in muscle, the default state of the phosphorylase is B, that is inactive, and the phosphorylase A is active. So here you get from the inactive state, you get to the active state. And it is activated by the AMP. So what means AMP? That you have a low energy state. AMP is much better predictor of the state than ADP for certain reasons, and most of the enzymes, they react on the AMP but do not react on the ADP. Well, because there's a lot of ADP around just from all that work, but only when AMP appears in some concentration, some high concentrations, you know that something is wrong, that you really desperately need for energy. So here is the slide that shows you the sort of phosphorylase kinase and the action of the phosphorylase kinase. So it activates by the phosphorylation. That's a massive protein, and that's a part of the glucagon epinephrine cascade. So here is phosphorylase A, once again phosphorylase B, and here you see that in these sort of cascades. Good. Just as a recap, so these signal molecules, epinephrine and glucagon, can bind to specific receptors. Epinephrine binds to the beta adrenergic receptor in muscle, whereas glucagon binds to glucagon receptor in liver. These binding events activate G protein, and then the cascade goes. So GTP bound subunit of G protein activate the transmembrane protein adenylate cyclase that synthesizes CAMP. The elevated level of CAMP activates protein kinase A. The binding of CAMP to the inhibitory regulatory subunits triggers their dissociation from catalytic subunits, and free catalytic subunits are now active. And the protein kinase A phosphorylates phosphorylase kinase first on the B subunit, then on the A subunit, which activates the glycogen phosphorylase. So that's the whole pathway. I'm not sure. Do I have an image here? Not sure. Good. I guess we are a bit scarce on time, so just dashing through to show you some more cool stuff at the end. So here is the once again glycogen and alpha 1,6 glycosidase. So here you see a little bit different bond. So we had considered the alpha 1,4 linkage, and here is alpha 1,6 linkage. So here is a more simplified scheme of whatever is happening. So here you see these are 1,4 linkages, and here is 1,6 linkage that gives you a branch. So phosphorylase cleaves alpha 1,4 glycosidic bonds. It cannot break the alpha 1,6 bond at branch points, so you need some different enzyme to do that. And it stops 4 residues away from the branch, so it cannot break everything it has, so it should stop somewhere, and it stops 4 residues away from the branch. And 1 in 12 residues is branched, so cleavage holds after something like 8 glucose molecules released per branch. And of course, we will need some different enzymes to perform something. These are so-called debranching enzymes. So there are further two enzymes. One is so-called transferase that moves a block of 3 residues to another chain. So here you see the action of the transferase. So I have removed all the glucose from this part. So I have removed that. Now my glycogen phosphorylase has stopped 4 residues before the branch. So here is the branch. Now what does transferase? It is able to transfer 4 of these glucose subunits to another branch to make it longer. So now once again on this branch I can operate with the glycogen phosphatase. And here only one subunit is left, and I need so-called alpha-1,6-glycosidase, the debranching enzyme that will hydrolyze at this point. So this is exactly the debranching enzyme. It removes the branch and it releases free glucose. So that's pretty different. When we were working on these sort of bonds, we released the phosphorylated glucose, phosphorylated at the first carbon atom. When we are removing this branched thing, we are releasing free glucose, and that may justify for you why do we performing all that transferring of these glucose subunits from here to here, because we want to get them phosphorylated. So we like phosphorylasis more than hydrolysis because we get already phosphorylated glucose and we can omit the step of spending 1 ATP for the phosphorylation. Here is the reaction catalyzed by phosphoglucomutase. Maybe I should have shown you these a little bit earlier. That's about the fact of having this phosphate on the carbon 1. So here you see what's happening. The reaction goes in a way that we have a phosphate attached to this serine in the phosphoglucomutase. This phosphate is used to phosphorylate at the carbon 6 to get our glucose 6-phosphate that we used to see in the glycolysis pathway, and then this phosphate is used to phosphorylate the serine back. So we are giving 1 phosphate to the glucose subunit, and then we get 1 phosphate from that subunit. Here are the details of this reaction. I think they are pretty interesting. So here you see the attack of these electrons on the phosphate and getting these phosphate for the glucose. Now you have glucose with 2 phosphates. It's a bit symmetric, a bit, because you have here a carbon atom, but you don't have it here. And it rotates around this axis. So around this axis it flips, and now you have this phosphate looking at serine, and it can be attacked with the electrons from the serine. This phosphate can be attacked, and then it's attached to the serine, and we see that 1 phosphate from the serine was transferred to the 6th carbon atom, while the phosphate from the 1st carbon atom was reattached to the serine. So glycogen is synthesized by a pathway that utilizes uridine diphosphate glucose, UDP glucose, as the activated glucose donor. So they should be activated before they go into the synthesis. And here you see these reactions, how they are coupled. Here you see a pyrophosphate, so that is hydrolyzed to 2 phosphates. That is basically driving the reaction forward. You will often see this thing in the biochemistry when you are driving some reaction forward by hydrolyzing the pyrophosphate, because that creates you two molecules and increases the entropy. So that's basically the entropic part that helps to make all that energies to drive the reaction forward. So we should couple these reactions to to get forward and to activate our glucose. Then glycogen synthase performs the synthesis. So here I'm showing you the uridine diphosphate glucose, so UDP glucose, and it's connected to the glycogen, and here you see glycogen had N residues, now it has N plus 1 residues. So we have two isoforms, one for liver, one for muscle and other tissues. They only can extend chains with more than 4 residues, so they require a primer. They belong to the glycosyltransferase family, and they are structurally homologous to the glycogen phosphorylase. As the primer, we have so-called glycogenin. That's the protein where we start the synthesis. It provides the starter chain primer for the synthesis, and here we have adding always these new units. Of course, we need to have all that sort of branching. So here you see once again the glycogenin and all that branched glucose subunits. And we need some separate branching enzymes that will create alpha-1,6 linkages. So after in linear chain is formed, we have, so here is the linear chain, it will be cut with the branching enzyme and transferred to create transfers a block, something around 7 glycosyl residues, and reattach them via the alpha-1,6 linkage in the interior position. So new branch point must be more than 4 residues away from the existing branch. So this part that is left should be more than 4. That's of course for we should keep synthesizing and adding more and more glucose subunits to this lower branch. And you might guess that this increases the solubility of the glycogen. That's probably what we want. Here are all the reactions that are written down and of the glycogen synthesis. So we need glucose 6-phosphate, we need ATP, we need glycogen that we already started to synthesize with N subunits, and we need a molecule of water. And at the end of the day, we get glycogen with one more subunit. We use up our ATP to ADP, and we have two inorganic phosphates. So here they were hydrolyzed into two. This pyrophosphate was hydrolyzed to get 2 phosphates. And these steps, they are performed with different enzymes like phosphoglucomutase that rearranges phosphate and prepares glucose for activation, UDP glucose phosphorylase that activates glucose. Of course, inorganic pyrophosphatase that hydrolyzes pyrophosphate and pulls the step 2 forward, otherwise it will not go. The glycogen synthase that adds glucose to the growing glycogen chain, and nucleoside diphosphate kinase that regenerates UTP for the reaction 2. Here is once again the signaling cascade that shows you what's happening here. So starts pretty similar, gets to the phosphorylase kinase, and here you see that phosphorylase B is transformed into phosphorylase A, and that starts destroying the glycogen and creating glucose 1-phosphate, and of course this cascade may be shut down, so that depends. So here you see the insulin that tells you what to do, so glycogen synthase kinase that works on glycogen synthase, and so on. I guess since we are so scarce on time, I will skip some parts. You can check regarding the hormonal control, and these are regarding the subunits that I have told you. So these are catalytic subunit and regulatory subunit, and how are they phosphorylated under the action of that phosphorylation cascades, and how that all works. So I will not get into that. Let's briefly get through different kinds of muscles that we have, and maybe consider something interesting in here. So except for skeletal muscles, we have smooth muscles and we have cardiac muscles. So here is here are some examples of smooth muscles. They are pretty diverse. They differ in size, organization, type of stimuli. They respond to some pattern of innervation, functional role, and so on. But they can be divided into two broad categories, like multi-unit smooth muscles. So here you see that on the left, and unitary smooth muscles that you see on the right. So here are some some examples of how do they differ. So it's what do I want to show you? But what is more interesting, probably the difference between smooth muscles and skeletal muscle. So they have no striations. So actin and myosin are not organized into sarcomeres. So we have seen sarcomeres, and here they are not organized. Instead, we have dense bodies. That's actin filaments attached to dense bodies. They are analogous to the Z discs that we have. So some are connected to the membrane, others inside the cell, and they are basically to transfer the force. Here you have a different filament ratio, and you have side-polar myosin filaments that's cross bridges on opposite sides that pull in opposite directions. And as a result, muscle can shorten up to 80% of its length. That's versus 30% in skeletal muscle. So it may shorten much more can shorten. Here we have some comparison. So here you see that smooth muscles are like slow tonic contractions, while skeletal muscles, they are more rapid and they perform some phasic contractions. Once again, I'll not get through all these details. You can check them after the lecture. So this is just some sort of comparison between these muscles. What I want to show you, there are certain biochemical differences. So in place of troponin, smooth muscle cells contain a large amount of calmodulin. Calmodulin initiates contraction by activating the myosin cross bridges. The calcium concentration in the cytosolic fluid increases as a result of the influx through calcium channels and or release from the sarcoplasmic reticulum. Calcium ions bind reversibly with calmodulin. So here you see calmodulin. Here it is bound to the calcium. The calmodulin calcium complex joins and activates the myosin light chain kinase, a phosphorylating enzyme. So here it is, and one of the light chains of each myosin head, called the regulatory chain, becomes phosphorylated in response to the myosin kinase and obtains the capability of binding repetitively with the actin filament and proceeding through the entire cycling process of intermittent pulls. So here you see it is phosphorylated, one of the two heads, and they become active and they start all that cycle. So that's pretty different of comparing to what we have seen in skeletal muscles. So take a note of that. The sources of calcium in smooth muscles, they mainly depend on the extracellular calcium, while in skeletal muscles we were mainly depending on the sarcoplasmic reticulum. And sarcoplasmic reticulum in these muscles is poorly developed. So that's interesting thing to notice. So more what else? Relaxation mechanism. So once again, we have calcium pump as we had in our skeletal muscles, but it removes to the it may remove either to the sarcoplasmic reticulum, that is a pretty slow process, or to the extracellular space or extracellular fluid. Because this is a long process, that explains the long-lasting contractions, in comparison to the skeletal muscles. They allow the prolonged tension with minimal energy use. That's pretty good. What else? there is a bit different pattern of the innervation of these muscles. So that's what I want to show you. The nerve fibers, they run along the membranes and of these smooth muscles, and sometimes groove their surfaces. The multiple branches of the postganglionic neurons are beaded with enlargements or varicosities. So here you see that varicosities of these varicosities are used to control the smooth muscles. So you remember that was pretty different from the skeletal muscles we have considered yesterday. And there is even some more differences that there are in the heart, but I think we will consider heart separately. So here you see neuromuscular junctions in smooth muscles. So here you see that sort of varicosities, and here you see the receptors of the smooth muscles. That's for the visceral muscles, and these are for the multi-unit muscles. So here you see they are pretty different of what we have seen for the skeletal muscles. So unitary muscle cells are connected by gap junctions. So so if we depolarize one of them, the depolarization can rapidly spread. In multi-unit smooth muscles, each cell is stimulated independently by neurotransmitter released from the closely associated varicosities. You can check a bit more information on that later. So here's some information on the junction, but I want to skip that. I want to show you, I still have 7 minutes, and I want to show you one very special muscle, that is the heart muscle. So here you see the heart. So I hope you remember a little bit something regarding its anatomy. So at least there are 2 ventricles and 2 atria. So here you see the right atrium, here is the left atrium, and here are right and left ventricles. The muscle in the heart, it is striated like skeletal muscle, contains intercalated discs. The connect cells through gap junction and allow ion diffusion for rapid excitation spread. Functionally, it works as 2 syncytia, so atrial syncytium and ventricular syncytium, and they are separated by fibrous tissue except for AV bundle. There are certain interesting things regarding the heart muscles, and here you see the action potential for the heart muscle. Since we have no time, I will not give you tricky questions. I will point that out on myself. Please look at this form, and you remember that the action potential we have considered, for example, for neurons or some other cells, they look something like this. So we need to go down, and then some hyperpolarization, and then go back. Here you see a long plateau. So we have this sort of plateau state. That's because we have one new player here, and that's the calcium. That's the first thing. The other thing, we have a plethora of different calcium channels, and some of them work pretty differently of what we already know. So in the depolarization phase, we have the sodium influx, so quick opening and inactivation of sodium channels. Here you see that influx on this image. So sodium channels have opened, some influx of sodium channels, and then they closed. Then we have we have some rapid potassium channels not shown in these plots, but they are responsible for these small, like going down this small step. Then we have another calcium channels that are almost always open, but when the cell gets depolarized, they almost stop letting any ions from the inside to the outside. But we have also the calcium channels that open, and we have some sort of balance. And during this balance, during the plateau phase, when we have almost no calcium efflux, that would have brought our potential back to the base level, but they are almost all the time closed. Instead, we have some calcium influx, and we have this long phase. So we are slowly getting back to the resting potential, membrane potential, but very slowly. And only at some point these calcium channels, they are starting to let the ions go out, and we have this third phase where calcium channels, they close, but calcium channels start letting out the ions, and we are getting back to the resting potential like -80, -90 millivolts. Here you see another image that shows all these parts. So here you see the numbering. It's pretty, I guess, standard. And here you see even more calcium channels. So here you see a sodium current, how it goes here, and some calcium channels, and here you see the transit outward current and different calcium channels like delayed rectifier rapid current, rectifier slow current, and inward rectifier current. So some of them are transient outward flow of calcium makes this sort of small step. Then we have plateau, that's opening of the L-type calcium channels. Then calcium channels inactivate, calcium flows out, and we are rapidly getting back to the state 4. That of course controls the excitation contraction system. So I don't think I will get into all the details. I'll just show you this image. So once again, we need calcium. This calcium comes from the outside or from the sarcoplasmic reticulum, whatever it means. And here are some more interesting notes. Here you see that we have certain cells modified in so-called sinoatrial node and atrioventricular node. And these two nodes, they basically define the heartbeats. So these are modified cardiomyocytes that are not neurons. Sinoatrial node, that's a pacemaker of the heart. It basically generates the heartbeats. Here you see the action potential in the sinoatrial node. There are some additional channels due to leakage in these channels. The resting potential is slowly getting towards the depolarization, and at some point they fire. So in this image you see the sinoatrial node fires these signals. The atrioventricular node just creates some sort of some time gap, so it gets signal, but signal propagates very slowly through this node, and so we have the situation when first the atria are activated, and only then the signal goes to the ventricles, and we have activation in ventricles. So here you see once again these plots and which ions are doing what. So slow influx of sodium makes us depolarize and get to the firing. So here you see the rapid influx of calcium, then efflux of potassium, and so on. Not getting into that, so here you see times that needed for the signal to get different parts of the heart. From the sinoatrial node to atria to AV node, and now through the AV node down the bundle of His, they are going down. And through all the heart. Here is, I guess, here you all know that this is the ECG. This ECG is basically you get some signals from all that depolarizations and repolarizations. So here you see how that looks like. But I want to skip up to this video, and I will show you this video, and that's probably the last slide that we are considering. So let me show you what's happening here. So here you see the ACG, and here you see the heart. The explanation is in this video is not really good, so I will explain on my own what is happening. So here you see the sinoatrial node. The sinoatrial node is a pacemaker, so it will generate you signals according to that process above. So here you should see the atrial depolarization. So the depolarization of atria, these depolarization spreads through all the atria. This is in the ACG, you see these as the P peak. So you see this P peak, then QRS complex, then the T peak. So this P peak, that's the depolarization that is spread through the atria. Starting from the P and plus, I guess, 100 milliseconds, we will get atrial contraction. So atria should start contracting and pump the blood whatever it needs to be. The PR interval from atrial depolarization to ventricular depolarization, that's the PPR interval. That's basically atrial contraction plus AV node delay. So here you see the AV node, that's atrioventricular node. It delays the signal that comes from atria. So you will see depolarization later. When the time comes, we will see the QRS complex. So that QRS complex, that will be ventricular depolarization. So this is the depolarization of the interventricular septum. Then you get the depolarization of the main mass of the ventricles. That will be when we get to the R. So here we are on the Q, so we are depolarizing the septum. So we went through the His bundle. Now we are on the Purkinje fibers. So we are on the R, and we are depolarizing the main mass of the ventricles. And on the S, that's the last phase towards the base of the heart, and the atrial repolarization is obscured in the this QRS complex, so we will not see that. So here you see how everything contracted, and now ventricles are fully depolarized, and so when we get to this sort of ST segment, so ventricles will be depolarized, and here we see the ventricular contraction and ejection of blood. And the T interval, that's ventricular repolarization. So now ventricles are repolarized, and here you see this sort of T peak. So that's how the ECG is compared to what we see in the electric system of the heart. So here you see how the blood goes and what's basically happening. So really nice video, but I guess there were some mistakes in explanations, so I have turned off the volume. Good. I guess my time is up. Sorry once again for getting too much of information. If you have any comments or questions, please go ahead. Okay, I guess no comments, no questions. Everyone's tired. Okay, good. Then have a nice evening and see you next week.